So this part of the presentation is on essentially the first part of the biology of the nervous system. And it basically starts off with the concept that all behavior is ultimately rooted in the nervous system. So if we understand the nervous system, we understand human behavior. Of course, the brain and spinal cord make up a huge amount of the nervous system, about 100 billion cells. You'll notice on the slide that 70 billion of them are dedicated to the cerebellum, which is involved in smoothing out movements. The microanatomy of the neuron is pretty basic. All 100 billion of them have three parts: the cell body, which contains the DNA and controls cellular functions; the axon, which takes information away from the cell body, and they can be pretty long, especially motor axons, 2 to 3 feet long. And of course, they're surrounded by a myelin sheath of fat that actually can increase the speed of conduction. Dendrites, on the other hand, are the third part, and they take information to cell bodies. And there's about 10,000 dendrites per neuron, which makes up 1 times 10 to the 15th connections in the nervous system, a staggering amount of connections. So we're born with all the neurons we'll ever have, although the synaptogenesis, that is creation of new synapses, happens throughout the lifespan. There are periods of time in childhood and adolescence where thinning occurs, that is, neurons that aren't used are pruned away. But the idea that some neurons can take over functions of others is fairly important, and that's called plasticity. You can see pictured in the slide the basic parts of the cell body: the axon, the dendrites, and the cell bodies. You'll notice too that the myelin has brakes in it. That is what increases the speed of conduction. It also is responsible for some demyelinating disorders like multiple sclerosis, where you literally get shorts in the nervous system. Different types of neurons stain differently with different chemicals. So you'll notice on the far left, the neuron in the cerebral cortex has many dendrites and takes a blue stain, whereas the spinal cord neuron has fewer dendrites, longer axons, and takes a purple stain. And then, of course, the retina has its own axons, which the optic nerve is actually retinal ganglion cells that come directly from the eyeball. And this is how histologists who study tissues know what type of tissue they're dealing with. The action potential is probably the hardest concept of all, and to get in the entire course. So you're welcome to read this over, but I'd rather show you on the next slide what happens. So the cell membrane is about 0.02 microns thick, and the gap between neurons is about 0.05 microns wide. So essentially what happens is, as the membrane opens and closes, it allows negatively charged particles like sodium to come into the cell. So let me show you this electrically, this action potential, on the next slide. So at resting potential, the axon, or the inside of the neuron, is minus 70 millivolts. When depolarization happens, the membrane opens up at about 1.5 milliseconds. It quickly jumps to a positive 40 millivolts. That's 110 millivolt change because of the sodium flooding in. At the top of the depolarization, the membrane closes, leaving sodium ions trapped in, the positive charge trapped in, so it's completely spent. At which point a sodium-potassium pump starts to pump potassium out. So sodium floods in by diffusion and is actively pumped out. Potassium is actively pumped out using energy. It pumps so well during this refractory period, which means it can't fire, that it actually takes about 5 milliseconds to restore itself. So if you think of it this way, the axon can fire up to, or the neuron can fire up to 200 times every second. So the delicate balance of electrolytes, especially sodium and potassium, is important. Incidentally, we get our electrolytes from many sources: sodium chloride in the form of salt and potassium chloride in the form of a stable element that comes from food. So we get salt pretty much from salt, and we get potassium from bananas, potatoes, leafy green vegetables, citrus fruits, and of course they regulate all the muscle and neuron activity in the body. You can see in this slide the propagation of the neuron across the membrane. As it depolarizes at 2 milliseconds, the wave spreads rather quickly, letting sodium come in and pumping potassium out. But when the neuron actually gets to the end of an axon, toward the dendrite, something happens. On the presynaptic side of the neuron are what are called neurotransmitters. They're stored in vesicles, and once the membrane opens up in the synapse, the vesicles float across the space. Remember I said it was about 0.05 microns wide. They start accumulating on the postsynaptic membrane, causing a chemical reaction that makes the membrane twist open. And once it twists open, it allows sodium to flood in, and another action potential occurs. It's easier to see on the next slide. So as the impulse approaches the presynaptic membrane, it releases, it opens the membrane up on the postsynaptic membrane, which causes sodium to flood in. This, incidentally, is where many pharmaceutical agents like antidepressants work at. They either work in the presynaptic membrane, the synaptic cleft, the space between them, or the postsynaptic membrane. Many of the neurotransmitters that we use in our body for muscle and nervous system activity come from our diet, which we'll talk about in a minute. What are some of the neurotransmitters? So they include acetylcholine, which is important in memory functions. It seems to be implicated in Alzheimer's. It's important in neuromuscular junction, so when nervous system innervates a muscle. There's a illness called myasthenia gravis, in which the person basically doesn't have acetylcholine in the synaptic vesicles, and essentially they suffocate. It kind of mimics the effects of some poisons like botulism poisoning or curare, which causes the blocking of acetylcholine release. Black widow spider envenomation does just the opposite. It causes the massive release of acetylcholine. And of course, the way to break acetylcholine down is to use an enzyme like acetylcholine esterase. If you ever see ACE at the end of a word, it means it's an enzyme. But it too is a protein. So you can be sensitized not only to the venom, but also to the antivenom. There's three neurotransmitters that are involved in depression: norepinephrine, dopamine, and serotonin. Norepinephrine seems to be implicated in the anxiety and physical symptoms of depression, like slowness or difficulty thinking. And so some antidepressants focus on norepinephrine. At least that's the theory. Either stimulating the production of norepinephrine or blocking the breakdown of it. Some are involved in dopamine as a neurotransmitter, which is involved in the mood aspects of depression. It also seems to be involved in movement disorders like Parkinson's disease, causing tremors or difficulty with soreness and stiffness or movement. And also involved neurotransmitter with depression is serotonin, which tends to focus on the cognitive aspects of depression, the negative thinking. So some drugs like selective serotonin reuptake inhibitors like Prozac focus on specifically enhancing serotonin in the postsynaptic membrane. When a person sees a psychiatrist for depression, typically the psychiatrist evaluates the types of symptoms they're having and prescribes medications based on their symptom picture. But if you know anyone who's just suffered from depression, knowing those things is no panacea because very often medications don't work. There are other ways to stimulate um, mood enhancement, by the way, and they include exercise and stimulating the brain to produce what's called more BDNF, which is brain derived neurotrophic factor. So, exercise, eating a good diet, sleep, stimulating learning activities, new activities are all ways to stimulate the production of neurotransmitters. There are endorphins, which are naturally occurring substances in the body that produce a natural pain killing mechanism. So after hours of exercise or work, person's not sore because endorphins finally, especially in the muscles, naturally blocks the pain impulses until the next day, of course, when your muscles are very sore. These also lead to problems with addiction. Anandamides, on the other hand, are found in marijuana-like substances. They cause a runner's high, perhaps, that and along with certain opioid receptors. Probably the two most prominent neurotransmitters in the nervous system are GABA, gamma aminobutyric acid, and glutamine, which is an excitatory neurotransmitter. So in a sense, think of the GABA as the brake pedal in your nervous system, slowing things down, and glutamine as the gas pedal, speeding things up. All the other neurotransmitters trim and guide the car, if you will, guide the nervous system. I just wanted to mention three other ones, including vasopressin, which regulates the fluids in your body as it's released from endocrine glands, oxytocin, which regulates the production of milk, especially during lactation, and N-methyl D-aspartate, NMDA, which causes a buildup of certain functional and structural properties within the synapse. So we know when a person learns something, there are actually structural changes that happen. Where do you get neurotransmitters from? From the diet, from meats and fish, from dairy products, milk, cheese, yogurt, from eggs, albumin. You can get them some from grains and vegetables, but per ounce they're far fewer than meat products. And of course, you can get them from beans and other legumes and grains.